A 7-day water-only fast, 12 volunteers, 3,000 proteins tracked daily. What researchers found changes how you think about fasting and when it actually starts working. Published in Nature Metabolism, this was one of the most detailed molecular maps of human fasting ever produced. Researchers measured approximately 3,000 proteins in the blood of 12 healthy volunteers every single day, before, during, and after a 7-day water-only fast. For the first 2 days, the body does what you'd predict. It switches from burning glucose to burning stored fat. Ketones rise. The expected metabolic shift happens, but the proteins associated with genuine health benefits, the ones linked to tissue repair, immune regulation, and organ remodeling, didn't move significantly during those first 2 days. The change happened at day 3. After 72 hours without food, the researchers observed a whole body systemic response, distinct from the metabolic shift that happens earlier. Over 1,000 proteins changed significantly across all major organs. 9 distinct proteomic response profiles were identified. 1 in 3 of the 3,000 proteins measured showed significant change. These weren't random fluctuations. The changes were remarkably consistent across all 12 volunteers, suggesting this is a conserved biological response, not individual variation. One of the most striking findings was in the brain. The structural scaffolding that surrounds brain cells, called the extracellular matrix, showed extreme changes in a protein called tenascin R, suggesting the brain's environment is being actively remodeled after 3 days without food. The implications of this are still being studied. The researchers linked the protein changes to genetic data from large population studies to estimate health consequences. One protein change was associated with a reduced risk of rheumatoid arthritis. That protein is called SWAP70. Another was associated with a reduced risk of heart disease, HU1. These are associations, not proven causal links, but they point toward biological pathways that fasting appears to activate in a way that shorter fasts simply don't reach. The study also identified potential adverse effects among the protein changes. Fasting is not universally beneficial at every level of biology, and the researchers said so. The finding that matters for anyone thinking about fasting is the threshold. The biology that might actually matter beyond weight loss, beyond ketosis, appears to require at least 3 days to activate. A 16-hour fast, a 24-hour fast, even a 48-hour fast may not reach the response profile this study identified. That doesn't mean shorter fasting has no value. It means the molecular response observed here is specific to extended caloric restriction. The first 2 days are not wasted. They are the runway that gets you to day 3. The science of fasting has a threshold. One study, 12 volunteers, 3,000 proteins. The threshold is day 3. If you found this useful, you know what to do.